Hi, I'm going to talk today about light regulation of enzyme allostery through photoresponsive unnatural amino acids. Specifically, the investigators of this 2019 through exposure to through light exposure of different unnatural amino acids. The unnatural amino acids are shown in Figure 1b, and this IMGPF protein, as you can see in Figure 1a, converts glutamine to glutamic acid and uses the freed ammonia to conduct other reactions at a separate active site. Other investigations in the past have regulated enzymes, and even some of them have regulated enzymes reversibly using unnatural amino acids, such as the AZOF, which you can see in Figure 1b, which is being used in this paper. However, what was significant about this paper is that with the exception of one previous study, no one else has done regulation of an enzyme through unnatural light-responsive amino acids away from the active site. All other attempts have been introducing these amino acids right near the active site to block it or otherwise foul it up, so to speak. In this study, they only made modifications of these unnatural amino acids at positions that were more than 10 angstroms away from the active site of the His-H complex, and that means that they're in the His-F domain of this multi-domain enzyme. The protein works in conjunction, these two proteins work in conjunction because the ammonia freed by the glutamine to glutamic acid is then channeled through the His-F protein to be used for other reactions. But molecular dynamics simulations showed some interesting results. So, first of all, the AZOF-Z conformation, which is the active version of the photoresponsive enzyme, makes the hinge region between the His-H and His-F rigid, preventing the flow of glutamic acid through the active site of the His-H subunit. Additionally, through some allosteric effects, the Z conformation of AZOF makes some backbone conformation changes, including a residue in the His-H active site in the other half of the enzyme, and it was previously hypothesized that this enzyme was important in the reaction mechanism because that specific residue was thought to stabilize an oxyanion intermediate. Molecular dynamics simulations that include the unnatural amino acids lend further credence to that theory because they show how these mutations affect the allostery of the protein as a whole. This is a really interesting study, and the authors hoped that they could further apply these lessons that they learned from allosteric control of enzymes to other enzymes that may be more useful industrially.